Regulation of heme synthesis. So what is the meaning of regulation? Actually controlling or maintaining the speed of the pathway so that it operates correctly. And regulation can occur either through activation of the enzymes or inhibition of some of the key enzymes, and also some drugs also have a role in regulation process. So basically controlling the rate of the pathway is called as regulation. So how heme synthesis is regulated? There are different mechanisms which regulate this heme synthesis: allosteric regulation, repression mechanism, compartmentalization of the enzymes, feedback inhibition, and there are some drugs which activate whereas which inhibit the heme synthesis. So firstly, allosteric regulation. So the word allosteric means other. Allos, the meaning of the word allos means other. So any site other than the active site is called as allosteric site. So inhibitors, if they bind to the allosteric site and if they stop the chemical reaction, that particular inhibition is called as allosteric inhibition. So this enzyme, key regulatory enzyme of heme synthesis, that is synthase, delta amino levulinic acid synthase, is allosterically inhibited by hematin. So Fe2 plus, if it is oxidized, it forms Fe3 plus, and this Fe3 plus is called as hematin. So if in the body there is excess of free heme, if there is excess of free heme, the Fe2 plus will get oxidized to Fe3 plus, which is otherwise called as hematin. Now this hematin will go and bind to the allosteric site of the enzyme synthase. So when it binds to the enzyme synthase, it will stop the heme synthesis to occur. So that is what is called as allosteric regulation. Synthase is allosterically inhibited by hematin. Hematin is nothing but when Fa2 plus is oxidized to Fe3 plus, that is called as hematin. So next type of regulation is repression mechanism. So repression is mainly occurs through the control of gene expression. So in the gene there are different areas: regulator gene, promoter site, operator site, and then structural gene. So during the synthesis of this particular synthase gene or enzyme synthesis, the excess product of the pathway shuts off the synthesis. So repression is mainly the mechanism by which the excess product will shuts off the synthesis of the key enzyme of the pathway. So if heme is present in excess, it will function as a co-repressor and it will bind to the operator site along with the aporepressor so that the synthesis of this synthase enzyme is stopped. So heme inhibits the synthesis of synthase, acting as a co-repressor. Now another type of regulation is compartmentalization of the enzyme. So the first step of the heme synthesis occurs in the mitochondria. The first step, that is succinyl CoA plus glycine, in the presence of the enzyme synthase, forms delta amino levulinic acid. This entire step occurs in the mitochondria. But the enzyme synthase gets synthesized in the cytoplasm and it is transported to the mitochondria for the reaction to occur. Now, if there is excess of heme in the body, the end product heme regulates the pathway by inhibiting the transfer of the enzyme from the cytoplasm to the mitochondria. So, heme will stop the transfer of the enzyme synthase from the cytoplasm to the mitochondria so that this step does not occur. So that is why synthase is a key regulatory enzyme. Now the another mode of regulation of heme synthesis is by feedback inhibition. Now what is feedback inhibition is when the product itself will inhibit the key regulatory enzyme, that is called as feedback inhibition. So heme will inhibit the synthase enzyme by feedback inhibition. And drugs which induce heme synthesis, as we discussed, some drugs will inhibit or will activate induce heme synthesis. So barbiturates, phenytoin and antifungal drugs, they require heme containing cytochrome P450 for their metabolism. So increased utilization of cytochrome P450 by these drugs will deplete heme, leading to the induction of allosynthase. And drugs which reduce heme synthesis are lead, mainly inhibits the enzyme ferrochelatase and aldehydatase and thereby inhibits the heme synthesis. It will inhibit this activity, ferrochelatase and aldehydatase, and thereby inhibit. And also another drug, that is nicotinic acid hydroside, which will decrease the availability of pyridoxal phosphate, which acts as a cofactor or a coenzyme for allosynthase and thereby affects the heme synthesis. So these are all the different methods of regulations of heme synthesis: allosteric regulation, repression mechanism, compartmentalization of the enzymes, and then feedback inhibition, and some drugs which are inducing and whereas inhibiting the heme synthesis. So main key regulatory enzyme of heme synthesis is allosynthase. This enzyme is regulated by different mechanisms. Thank you.